And it's my pleasure to interview the next speaker, Clemens Wittenbecher from Sweden. And the topic is Metabolomics, Mendelian randomization, and other multi-omics approaches for dietary assessment. Clemens, the floor is
okay, you have the pointer. Yeah, okay. Yeah, thanks for having me here, and it's a pleasure to talk about causal inference in nutritional epidemiology. And I want to start by just referencing the updated guidelines on fat intake by the WHO, which basically recommend what most of us would also recommend: reducing saturated fat and replacing it primarily with plant-based unsaturated fats, polyunsaturated fats. But I also want to highlight that the only high certainty evidence, despite decades of research on that topic, they consider is LDL cholesterol reduction. For this reduction of cardiometabolic diseases or all-cause mortality, the evidence is still considered low to moderate. And then they, in parallel, last year, the guidelines on amount of fat were released. And the target amount is 30% in relation to weight reduction or prevention of weight gain. And this is maybe more controversial here. I think we just heard that low carb diet is a great way to reduce weight. So, but again, a conditional recommendation, so it's not applying for all. So the question is, despite these decades of research on the cardiometabolic risk effect of fats and different types of fats and fats in comparison to carbohydrates, it's still not considered conclusive evidence. And I mean, this is a review by Nita Faruhi a couple of years ago, showing just that there has been quite an evolution of our understanding how dietary fat affects the risk of developing cardiometabolic diseases, while a few decades ago, all the attention was on the effect of cholesterol or lipoproteins. Then now we understand that this is part of it, but we would like to also understand in more detail how different lipoprotein fractions are affected differently by type of fat. And on the other hand, we also assume that there are quite a few non-lipoprotein mediated effects of the dietary fat on cardiometabolic risk through, for example, membrane composition, lipid signaling. And if we look into this very broad definition of precision nutrition, this is from the NIH, the research framework for their nutritional research agenda for this decade, and defining four main goals, from foundational research over the effect of dietary patterns, nutritional effect across the lifespan, and then the goal to implement that. But this is then really also pointing into a more thorough understanding of the causal mechanisms or the causal processes that connect dietary effects or dietary factors with disease risk. And it appears that despite that we have a lot of evidence actually for fat intake, we're still not there. So what I will talk about mostly is different approaches, how we have tried to understand that process of how the composition, specifically for fat, mostly in that presentation, affects the metabolic adaptation, the longer term metabolic adaptation, and how that translates into different cardiometabolic risks. And in the first set of slides and results, I will focus on modeling data-driven metabolomics networks and applied to like the group of dihydroceramides and ceramides. I think many of you know that this is like a group of metabolites that is very interesting metabolically because it strongly responds to dietary interventions or is associated with dietary intakes in observational studies. And it's also very relevant for disease risk, both in relation to type 2 diabetes and cardiovascular diseases. So it's an interesting group, but it's also clear that within this group of ceramides, we have like a quite diverse composition of fatty acids within the ceramides. And we wanted to understand like how specific foods that are fat either influence lipid metabolism or fat sources in the diet influence like how differently affect the lipid composition within the ceramide. And to this we use the algorithm to like create a metabolomics or metabolite network and identify the most like robust factors that respond to diet and are associated with disease risk. We call that the net coupler algorithm because it kind of couples exposures and disease risk to metabolomics networks. And what we like, the main assumption is that we want to control within a network of disease-related metabolites or lipids in that way. We want to identify the factors that are directly influenced or most likely candidates for being on a biological path. Because as you know, like they are all highly correlated, and we want to really depropagate the associations with both diet and disease risk and zoom in on the most relevant factors. And for that, we use basically conditional independence theory so that we can, if we have a conditional independence network, if we control for the direct neighbors in such a network, we can control for all influence of the whole network. And since if we want to really have like that as a causal modeling framework, we would need to know which are the direct neighbors that affect the metabolite and which are affected, but we don't have that. So what we do in this net coupler algorithm is that we basically estimate all the network adjusted models and zoom in on the metabolites that are associated either with disease risk or with the dietary exposure, independent of any adjustments for direct neighbors. So it's the ones that are really like independent of any network adjustments associated with type 2 diabetes risk are the ones that we consider as most interesting. And here we did that for the network of ceramides and dihydroceramides and identified a group of ceramides that, independent of any adjustment for other ceramides and dihydroceramides in that network, are stably associated very substantially with an increase in type 2 diabetes risk, and another group with risk of cardiovascular diseases. And we then linked this network with diabetes and with red meat intake, really focusing on these metabolites, these that are network adjusted, robustly associated with type 2 diabetes, looked into the relationship with red meat intake. And what we found here is that we do that in a mediation modeling framework, that the fatty acid composition in these group of ceramides that are really robustly against network adjustments associated with type 2 diabetes risk explains quite a large proportion of the red meat intake associated type 2 diabetes risk. So over 60% in the mediation model. It's still an observational analysis, but I think it is like from a biological perspective that, for example, one of the main effects is increase in the steroid concentration within ceramides is one of the major potential explanations for the higher type 2 diabetes risk in relation to red meat consumption is interesting hypothesis that can be further explored. And that's actually the only slide I have on Mendelian randomization, but anyway. So what we also did is to follow up these results up with other causal modeling techniques, including Mendelian randomization, and also found evidence for a causal effect. I think there are now other studies that corroborate that in larger sample sizes, that specifically ceramide 22.0 has a potential causal effect on type 2 diabetes risk, according to Mendelian randomization study. But then, I mean, for both the network adjustments, but also and mediation analysis, but also for Mendelian randomization, it still depends quite heavily on the set of like causal assumptions that you make when you go into these studies. And I think it is like what Vasanti was referring to, this combination of studies has a great potential to actually combine studies in such a way that you basically, through the combination of studies, exclude some sources of bias in the single study designs that are not easily controlled, even if you combine several studies of the same design. And this is something that I'm very interested in at the moment. So here are some results from this multi-study integration on the lipidomics imprint of replacing saturated fat with plant-based unsaturated fat. So mainly dairy unsaturated fats with plant-based unsaturated fats. And this is before the background that we actually, if we look into the older trials that look into this replacement in an actual intervention setting, we would expect a quite substantial CVD risk reduction that we don't necessarily extrapolate. We have very stable reduction of non-LDL or non-HDL or LDL cholesterol by this intervention, but it doesn't quite reach the target reduction that would necessarily inform such a substantial risk reduction. And what we then did is that we basically used this model where we have like the diet composition that we can relate in a temporal context to cardiometabolic disease risk in a cohort study, but then associating diet composition with markers of metabolic adaptation, so metabolomics or lipidomics, in a cohort study is much more prone to confounding than this longitudinal temporal relationship. So what we intended to do in the results that I will show in a minute is to use to link the diet composition to metabolic adaptation markers in a randomized controlled trial design, and use a larger set of metabolomics or metabolic adaptation markers, specifically for this saturated with unsaturated fat substitution, a set of lipid metabolites, lipidomics. And what we found is that a combination of the lipid metabolites that are significantly affected by a dietary intervention that really does that in a very controlled setting in the DIVAS trial, replacing saturated fat, 8% of total energy, with unsaturated fat, has a very strong effect on the combination of lipid markers, lipid metabolites. And that if we use this combination of lipid metabolites to extrapolate the effect of this diet intervention on long-term cardiometabolic disease risk, we actually come quite close to this expected estimate of about 30% reduction of cardiovascular disease risk, and that at least in the analysis that we did in the EPIC-Potsdam cohort, this is scaled to the intervention effect that was achieved in a trial, and then associating that with disease risk in 5 minutes. In a cohort, in the EPIC-Potsdam cohort study, and if we do exactly the same with non-HDL cholesterol, this extrapolated disease risk effect is much smaller. And then you refer to diet substitution modeling. If that is also interesting, that if we use diet substitution models in the nurses' health studies, modeling the replacement of saturated with unsaturated fat, we see a very similar effect on this multi-lipid score that combines the levels of several diet-affected lipid metabolites. We see that this multi-lipid score is associated with dietary patterns in the expected way. So this is animal-based low-carb diet, and this is actually a disadvantageous association, so lower level of this multi-lipid score that reflects a healthy diet composition, whereas a vegetable or fat or plant-based fat low carbohydrate diet score has a very beneficial association with this multi-lipid score. And again, we can associate these lipid scores with disease, replicate the disease risk association that I had shown for EPIC-Potsdam in the previous slide with type 2 diabetes, and also the change models that you referred to, so that I think we can be quite confident that these are very solid markers of the metabolic adaptation to a different fat composition in a very controlled setting, and that these are very robustly associated with risk of CVD and also type 2 diabetes. And we're working on similar analysis integrating low-carb diet intervention, actually run by David Ludwig in a collaboration with Johann Hirschhorn's group. So this is something you don't necessarily observe in free living populations, so 20% carbohydrate and 60% fat ratio. These two are more common, but in the trial we had like these three groups: a run-in phase with weight loss, and then a weight stable intervention phase where participants were randomized to these three diets. We see over 100 metabolites that are affected by this dietary intervention. And then we use, again, the nurses' health study that has this like more naturally occurring distribution of fat to carbohydrate ratio between 40-40 and 60-20. And what we see is a much more complex picture than for the saturated fat and unsaturated fat comparison. So, I mean, you see that quite a few of the metabolites that go up with higher fat versus carbohydrate intake are associated inversely with type 2 diabetes risk, but then we also see some high risk associations. So what we have in the prospective cohort study with that substantial sample size in the nurses' health study, we have the freedom to look into more detailed dietary substitution models. So if here we have the total fat substitution, and with higher fat and lower carbohydrate consumption, like all the triglycerides, for example, tend to go down. But then if we look into more specific monounsaturated, polyunsaturated, and saturated fat substitution models, we see that the polyunsaturated fat specifically reduce the triglycerides with low carbon chain length and low desaturation number, whereas the saturated fats specifically reduce the triglycerides with longer chains and higher desaturation number, which is expected, but it's also confirmed by the data. And if we look into the diabetes associations, for example, these are specifically associated with a very strong increase in type 2 diabetes risk. So that's the last slide. So I think our results confirm the cardiometabolic health benefits of substituting high quality unsaturated with plant fats for saturated animal fats. I think it's also, yes, I think it is a much stronger approach not to talk so much about hierarchy of study designs, but a way to combine studies so that that we have through the combination the possibility to address sources of bias more robustly than with any single study design.
Clemens, thank you for your really wonderful presentation.